No, thank you for the invitation and for the organization. I have had a lovely day talking with many of you here. And today I'm going to talk to you about what we do. The title of the talk is Bioenergetic Evolution, the link between ours and life's history. And we are mainly interested in different aspects of microbial evolution, but more focus on how microbes interact with the environment, how they produce ATP, so energy conserving strategies, and how these energy conserving strategies evolve from one to the other one. And I can say that the group has mainly three focus. The evolution of the redox modules that exist in bioenergetic systems, creation of tools to aid into the easy classification or functional annotations of protein data, as well as, because bioinformatics only gives you predictions, we are expanding either with collaborations or also at the University of Vienna to do characterizations of some of our predictions. But today I want to talk to you about life and how diverse life is. And there are currently around 2.2 million species identified so far, but the prediction is there are much more species available. And we also know that regardless of this diversity, regardless of different colors, different shapes, more complex, less complex, all of these life forms once evolved from one common ancestor, LUCA, or the last universal common ancestor. And since in the modern era, since we can do RNA trees, these types of phylogenies were created. And with the discovery of the archaeal domain by Oos and Fox in 1977, we have this picture of the three domains of life with bacteria, archaea, and eukaryote as a sister clade of archaea. And the same type of topology was retrieved using more sophisticated methods, such as concatenation of universal, mainly ribosomal proteins. And what these three tell us is the three domains of life, three with eukaryotes as a sister group of archaea. But more or less 10 years ago, there was a revolution in deep phylogenetics. And more independent groups start to retrieve phylogenies with the different relationships between the three domains of life. And in 2015, 10 years ago, by sequence metagenomic organism, it was discovered some archaea that are closer related with eukaryotes. And in these new phylogenies, we always got eukaryotes not as a sister clade, but branching inside of archaea, methanogens tending to be at the root of the archaea. Methanogens is an archaeal lineage. And Asgards, they gave it the name Asgards because the first lineage was a sequence near the Loki castle. And it was called Loki Archaeota. And then when they start to find more, this group is called the Asgards as the closest relative of eukaryotes. And this proximity, it's not just in terms of the phylogenetic trees of life. It's also seen by the presence of many ESPs, and ESP stands for eukaryotic signature proteins, proteins that were considered to be characteristic of eukaryotes that are in this lineage. You can see in the top line the balls, the field colors for the eukaryotic ones. And we can see that in low hierarchy, many of these columns are also filled. So this kind of tipped a long discussion and a long debate that existed about the 3 versus 2 domain of life. And now the more accepted view is that the eukaryotes are derived from archaea and bacteria, so they are younger than these 2 domains. And when we think about our last universal common ancestor, eukaryotes are not so fundamental and should be excluded. And when we want to look into the past and in such long distances and think about the evolution, we always have to have a bit of several disciplines. We, of course, we need biology and we need informatics to do all the bioinformatics reconstructions. But sometimes we also need philosophy to fill in the gaps and create scenarios to explain what the data is telling us. And also we can need geology to have some more information about our planet back then. And as you can imagine, this leads to many hot debates and to many long, decade-long divisions. Because since we have to start introducing philosophy and our ideas, we subjectively pick the arguments or the biological facts that we think that are more relevant, and someone else will pick another fact and propose a different scenario, and this leads to many heated discussions. And there is only one reason for all of these discussions, and it is we cannot go back in time. If we could go back in time, we could have seen exactly how things happen, but we cannot. But we can try to fill these gaps that we have by learning from other disciplines. For instance, so far, we as biologists cannot even agree on a definition for life. And the idea is to try to find constraints or things that all life forms share instead of giving a definition. For this talk, and I apologize if you don't agree, I'll give my favorite definition of life, which is life is the result of energy releasing reactions. It's an out of equilibrium system that it's only in equilibrium when we die. And with this in mind, I want to introduce you to one of my heroes, which is Peter Mitchell. And Peter Mitchell has done amazing work regarding bioenergetics, which is the study of how energy is transformed in a living organism, and created a concept that then proved true of chemiosmotic coupling. And this is something that happens in the majority of the cells of all living organisms, where we have a reduced donor that gives the electrons to some complex in a membrane, and these electrons will pass through some carriers in the membrane until they reach other complexes. Some of them, but not all, will pump or translocate protons from the inside to the outside of the cell. And this will make a difference in the concentration of the proton. And finally, the electron will go to a terminal complex and will be passed to an environmental available acceptor. We have a similar process happening in our mitochondria. In our case, we use oxygen as terminal acceptor. In the case of microbes, the diversity is much larger. And in metabolism in general, but especially in bioenergetics, there are some other properties that make it modular. And we can have this modularity at different levels. We can have the same reaction being performed by complexes with completely different evolutionary origins. And in here, it's an example of complex 1 that can be replaced in some prokaryotes from NQR or type 2 NADH. We can have a Lego organization of new complexes because life forms don't invent everything from scratch. Sometimes they reuse what they have and they just organize it differently, and we can have different complexes such as soluble hydrogenases or the MRPs antiporters that share common blocks by the same color with complex 1, and each one of them have different perform different reactions. And especially in what regards bioenergetics, at the core of all of these complexes, we tend to find metals, coenzymes, and cofactors. And even here, there's a small set of possibilities that are used and reused sorry, by the microorganisms over time. And we also know that biochemistry and geochemistry have been co-evolving for a long time. And in the words of William and Frausto de Silva, all the changes in the original life forms depend on energy as well as material captions and flow. So if we want to think in early evolution, what could have been the metabolism of our last universal common ancestor, we have to think what was available and what type of energy disequilibrium was being used to keep the system out of equilibrium. Because we have to keep in mind that enzymes per se do not invent a new reaction. They act as catalysts and make it faster. So if we think about Earth history, we know that our planet is around 4.5 billion years ago, billion years. And that we have indications that the first rocks and the first and water was in our planet around 4.2. And regarding evidences of life, the geologists debate. The 3.8 billion years ago is the more sound proof, although there are older at 4.1 or at 3.95, but those are still under debate. And our planet was not this blue and green ball that we have there. Our planet had no life in the beginning. And you can imagine that there was no ozone layer to protect from the radiation. And volcanoes were at the maximum of their activity. There was also meteorite showers coming into our planet. So it was a very harsh place for life to first originate. And according to geologists, there was almost no or even no oxygen for the majority of the time. And in here, you can see, you can see the log of oxygen across the years. And you can see that with the exception of some whiffs of oxygen before 3.0, the increase in oxygen in our atmosphere occurred later between 2.5 and 2.8 billion years ago. So that's when um, oxygen was available for the life forms that exist to respire. And while many geologists cannot tell us that there was no oxygen before, biologists in its majority thinks that this corresponds not to the appearance of cyanobacteria, but to the expansion of cyanobacteria in our planet. And we have evidence that oxygen has a much more peripheral role in metabolic networks. In this seminar paper, what you are seeing here is a combined network of all of the metabolic reactions possible by the organisms known at the time. And in red represents oxygen-dependent reactions, and in blue, oxygen-independent. And we can see that towards the end of the networks, we see much more red than blue. And in fact, when we look to the impact that oxygen-dependent reactions had in a microbial physiology, and here you can see a heat map where each row is genome. They are kind of organized by taxonomy. On the top, we have archaea. On the bottom, bacteria. And then each column represents some type of enzyme that belongs to a different category, such as metabolism of amino acids, aromatic fermentations, and so on. And we can see that oxygen was used either to replace some of the hard reactions that existed. For instance, in many cofactors biosynthesis, the cofactor was duplicated and the oxygen version, dependent version appeared, or to make reactions that were not so favorable in the anoxic environment easier, such as aromatic degradations. But we can think that oxygen was very good and allow for obtaining much more energy. But we have to keep in mind that oxygen appearance and incorporation in biological networks also came to a cost. And the cost is life is far more energetically expensive with oxygen than without it. And if we look to how much it costs to produce the building blocks of life, proteins, bases, and so on, we see that it's more expensive to build them up when oxygen is present. And this leads to the conclusion that first organisms, the ones that were already alive before the appearance of oxygen, were anaerobic. And you might think, well, that's obvious. You said that there was no oxygen before, or almost none. You have no idea about the debate that exists at the moment, putting oxygen-dependent metabolism at the origin of life. We can really debate everything. But if you believe me, or if you think that my reasoning is logical, by knowing that the first organisms were anaerobes, we can do some logical considerations. So if oxygen appear in our planet for biological usage due to the action of cyanobacteria with oxygenic photosynthesis, we can say that aerobic respiration is a process that is younger than oxygenic photosynthesis. There was no oxygen, so there was no aerobic respiration. And all the data points that an aerobic photosynthesis, the one in which the microorganisms only have one of the reaction centers, and there's no oxygen produced, is a process older than oxygenic photosynthesis. And one thing that is characteristic of all photosynthetic organisms is they all have chlorophyll, with no exception. So if an oxygenic photosynthesis was present, we had to have chlorophyll. And chlorophyll, it's biosynthesis. It's just a few steps from heme. You have to have a heme protein, which is a cofactor, in order to build chlorophyll. And since we are talking about hemes and phenols that are also needed for anaerobic photosynthesis, we can think that the first forms of respiration were of the anaerobic type. But in here is where we start being able to do more logical considerations, because there are many different types of anaerobic respiration in microorganisms. So we have to turn to another way to get more information. And there are mainly two different ways to look to early evolution. And by early evolution, I mean things that happened more than 2 billion years ago. One is geology, and the other one are the genomes. And in the words of a geologist, Earth records its own history. However, due to plate tectonic movements and erosion, part of this history has been erased. On the other hand, we know that there are still some records of microbial activity in old rocks. They don't tell us to which group they are affiliated with in the systematics or taxonomy, but they tell us what these microorganisms were doing for a living. And on the other hand, we have genomes that also record their own history. But similar to what happens in geology, we have lateral gene transfers and sequence divergence that is been erasing much of the evolutionary signal from the primordial organisms. But we can still see some of this history there, as long as we take into account that lateral gene transfer decouples the evolution of this 16S RNA from the evolution of the metabolism. And the connection between these two is microbial bioenergetics, because that's what is imprinted in the rocks. So to keep in mind, we always, when we look to evolution of metabolism, there's no much point of looking to the evolution of the ribosome. We have to look into the evolution of each one of these individual genes. And there are several traditional ways to try to reconstruct the genome of or the genes that were present in our last universal common ancestor. One was if it's present in both or in the three domains, should be present in Luca. But this, it's not much used anymore. And then we have a system in where we have a species tree or a reference tree. And then we have a presence absence matrix in that square rectangular in the middle. Black means that the gene is present, and white means that the gene is, or protein family is absent, with some model for gene gains and losses. And then depending on the criteria that we choose, we can get the 30 to 100 nearly universal proteins, or we can put everything back into Luca. And this strategy is highly dependent on the data set, on the reference tree, and on the model parameters chosen. But more important, this approach has a conceptual flaw. Anyone wants to have an idea, say something about it? It's true that plants have bigger genomes than animals. And here we are talking about bacteria and archaea. Some have smaller genomes, some have bigger. Most of them, I would say, are much smaller than the eukaryotic ones. But even if we would be comparing plants and animals, we would still be looking to the presence and absence of a gene. Potentially for plants or animals, this method would work. But it doesn't work for microorganisms, for bacteria, and for archaea. And it doesn't work very well because the assumption behind this comparison that we are doing is that each one of those genes have the same evolutionary history of the ones used to build the reference tree. And this is not the case because of lateral gene transfers. It's like someone, you can have easily in archaea bilateral gene transfer an entire trait that so far that originated in bacteria, and it was only present in bacteria, but it was transferred in there, in the blacks and whites, it will be the same dot as the other ones. So we decided to look at this in a different way. And we decided already a while ago to look for ancient genes using these two criterions. Since we are only using archaean bacteria, the genes should have been present in both. And we would do phylogenies, and we should still see a separation between all the proteins in archaea in one clade and all the proteins in bacteria in another one separated. And then to try to eliminate some recent lateral gene transfer, we would have to have at least 2 archaeal groups and 2 bacterial groups to try to eliminate potential recent lateral gene transfers for just one organism. And we picked around 2,000 complete genomes, genomes that were high quality, closed, and we did all the um, similarity searches, global identity, all versus all, clustering, and we obtained around 12,000 clusters or protein families, and we did this phylogenies, maximum likelihood alignments and maximum likelihood reconstruction of all of these trees. And then out of those, there were 355 only that still pass our filtering of archaean bacteria being monophyletic, one clade for each, and at least with 2 groups. And before continuing, because all methods have caveats, I want to spell it out. First, this method is obviously dependent on the data set used with different genomic data set. We will possibly not have exactly the same result. Then we might have in our clustering separated into sequences that were orthologous, that had a common ancestor, that are present in archaean bacteria, but because of our cutoffs and because of they being old and divergent, they split in different clusters. And we can also talk about some false positives. We found 8 out of the 355 proteins related with oxygen, at least from the annotations. And we also got some false negatives, because although some parts of the pathways were found, not the completely pathway was found. And because around 83% of these families have had some sort of functional annotation, we could try to group them by functional categories, as well as try to understand what would these families tells us about the metabolism and habitat of our last universal common ancestor. And in here, you can see a scheme depicting what the major findings, what we found. So in this analysis, we retrieve that LUCA was anaerobic, that's dependent on hydrogen and CO2. We also found some evidence for lipid, amino acids, and nucleotide biosynthesis, and as well as evidences for ATP production, both at the level of substrate level phosphorylation, as well as at the level of the ATP synthase. And all of these proteins, the chemistry and the cofactors they had, were based on metals, some specific cofactors, and many of them involve methyl-based chemistry. And more important, we found that part of the acetyl-CoA pathway was conserved. And the acetyl-CoA pathway is a carbon fixation pathway that is found in both archaea and bacteria, and that can be divided into different parts. In bacteria is found in acetogen, and this line over here, you can see the methyl branch of acetogens that start in CO2 and ends in methyl. And you see that many of the steps involve H4F, and this stands for folate, and folate is a universal methyl donor in all of bacteria. On the other side, we have the methyl branch of the methanogens, of the archaeas, and the transformations at the level of the carbon are very similar, but in this case, instead of having folate, methanogens have methanopterin, MPT. And in the center, we have the common part between both pathways, which is the acetyl branch that is performed by CODH or ACS acetyl-CoA synthase that is found in both, that is exactly the same in both acetogens and methanogens. And interestingly, this pathway has actually geological similarities with a process that happened in hydrothermal vents, which are kind of formations that exist, for instance, in the bottom of the ocean, but not just, possibly also in Enceladus, which is a moon from Saturn. And where even nowadays we see independently of life, so just chemistry, abiotically, methane being produced from CO2 and hydrogen. And this is in agreement with one of the theories for the origin of life that was proposed by Mike Russell and William Marty, and that states that due to the similarities of geology and also biology, that the acetyl-CoA pathway should have been the first carbon fixation pathway, and with methanogens and acetogens as living fossils of the early metabolism. So in here, you can see what was done. It was already 9 years ago, but there was a second attempt, or there was a second, not attempt, there was a second reconstruction of LUCA content from genomes. And this one is from last year, and it's represented here. And although the data sets were different, and what was common, and the methodology not exactly the same, is that this pathway, the acetyl-CoA pathway, was also found at the basis being present in LUCA. However, because it was different groups performing the analysis, the interpretation was also different. But phylogenetically, what this can tell you, it's that both methanogens and acetogens, one archaeal lineage, one bacterial lineage, their metabolism is ancient. So this was to show how exactly the same data can be interpreted in completely different ways. But today, we went from 1 genome per year to around 3,000 per day. Currently, there are around 3 million genomes, and the number of unique sequences is around half of the world's population. So with this amount of data, doing all versus all comparisons, it's not feasible. The data has different levels of, it's very heterogeneous. Some are more complete, some are more contaminated. And this leads to the use of protein sequences because it's much faster, it requires less storage, and also we don't have problems with third base variations. And we need better ways to deal with the data. And if you remember, in this scheme, we stopped in the logical about which one of them could be. And archaea and bacteria are active partners in many of the biogeochemical cycles, such as the carbon, iron, oxygen, sulfur, and so on. So we could start thinking that it would have been from one of those. Can eliminate oxygen and we started to look into the sulfur cycle. And the sulfur cycle in here, it represented all of the different transformations that microorganisms, bacteria in gray, archaea in orange, and that both domains of life know how to do in red. And you can see that through time, organisms have evolved and learned of ways how to deal with all of these transformations with the goal of building ATP. And we start with the dissimilatory sulfur metabolism that is present in both archaea and bacteria in several cultivated organisms. And there are geological evidences for a 3.5 billion years old sulfate pool. So sulfate would be available early on the history of life. And this is a very complex pathway. I'm not going to go over all of the details, just quickly. When sulfate enters the cells, and when we are in the direction of sulfate reduction, it's the red arrows. It will pass through a set of compounds until it finally is transformed into sulfide, S2 minus. And in this pathway, central to this pathway is the DSR-AB protein that together with the DSRC are responsible for the transformation of SO3 to minus to sulfide and to delivering then the electrons to the membrane for chemiosmotic coupling. And in the reduced direction, we have the DSRD that is only present in reducers. And then if we follow the blue arrows, we can go with the same complexes working in reverse to do sulfur oxidation. And for sulfur oxidation, it's thought that the DSR E, F, and H are characteristic and specific of this metabolism. But some organisms don't do the full conversion of sulfate and start with a more reduced SO3 to minus 4, but the rest is the same. And there are many ways to look when you have a genome to try to identify if the organism performs is a reductive type, is oxidative type, and so on. And this way is to build a phylogeny. Because these trees in here, it's representing DSR-A and DSR-B type, have always the same topology. We have Morella thermostatic second copy that many times is used for rooting the tree. Then we have the reductive archaeal type that is followed by a separation between the reductive bacterial type and the oxidative type. And how do? You have your DSR sequence, you do an alignment, you build a tree, and you see where it is. But this is very time consuming. So we decided to develop a tool to identify sulfate producing microbes in red. When we're talking about sulfate producing microbes, it's always red, and sulfate oxidizing bacteria in blue. And this tool started by looking to the literature, seeing what is known, what are the proteins that are characterized, and select some queries and some complete genomes to then do all the global identities, clustering, phylogenies, synteny analysis, looking how all of these genes and these pathways are organizing the genome, doing network analysis, as well as orthologous to paralogous distinction. And this leads us to a minimal set with DSR A, B, C, and K that is co-distributed with some co-distributed proteins, some of them very important for the process, as well as other additional DSR proteins. And part of the ortholog paralogous distinction was done in different ways. Here is an example of DSRC. DSRC is a protein that has around 120 amino acids, so it's a nightmare for a bioinformatician. It's very small, and it has homologous with regulatory proteins as well as a TUX-E. And the difference is, while the SRC can have 2 or only 1 cysteine, the RSPA has none and TUX-E has 1. So we could not even separate them by the number of cysteines, but we managed to find a way. And this leads to the creation of the tool that is based on hidden market models. Creating hidden market models is easy. Finding the cutoffs for the models was the hard part. And then it can use as input any sequence and gives you output identification of the protein as well as its predicted type. And we also did statistics. We created DISCO, and DISCO is very fast, takes around 3 seconds per genome in a normal computer, and it has an accuracy of around 100% when we compare it with our predictions with the literature phenotype. And it was good to have DISCO, and because what we wanted was to see the evolution of this metabolism. And when we started, there were 3 different scenarios for the appearance of DSR dependent sulfur metabolism. Mostly, not just, but mostly mainly on DSR A, B phylogenies. This metabolism has been proposed to be in LUCA or to have appeared very early in either archaea or bacteria. More recent, with the discovery of new lineages, the origin of the pathway was said. There were so many lateral gene transfers that it was not possible to determine anymore. And then in 2020, 2022, there was the proposal of an origin in a late lineage of archaea. In all of them, the sulfate reduction that was seen in Archaeoglobalis was the result of lateral gene transfer from bacteria. So we combined DISCO as long because we wanted to add additional proteins that were not covered in DISCO with extra similarity searches. And we had our data set with around 200,000 metagenomes. And after all of the filterings, we had around 50,000 hits that were then cluster aligned. Phylogenies were done, similarities, networks were done, and then we had the results. So when we look to the distribution, and in here you have a heat map where, oh, it's late, where in red is the presence of reductive type proteins, in green mixed ones, not all proteins have a type, or in blue in oxidative. You can see that we see more red than blue, meaning that there are more lineages, or the diversity of the reductive type is larger than the diversity of the oxidative type. And when we did all of the phylogenies of the DSR A and B, we found a topology exactly the same as the ones that had been published by everyone. The difference started when we looked to the other genes, and we start to see that the organization between the different clades was different, and in some organisms, instead, although being reducers and cultured in the lab, some of them were clustering with being oxidizers, sorry, were clustering with reducers and not with the other ones. And by our analysis of the lines that were basal, we got a similar result. Archaeal tends to be basal in all of the phylogenies. And this led us to propose a scenario in which there is an archaeal origin of the minimal set to do to transform SO3 to -2 sulfide, that then with the acquisition of more proteins got more complex, and only later on with the adding of KMO complex as well as the APR A and B was able to do the full reduction of sulfate to sulfide. And due to losses in genes and acquisition of others, the pathway was able to invert, and finally sulfite oxidizers appear. So there's an increase in complexity from simple to more complex, and with the reuse of some of the building blocks that were existing, that were changed the direction, or that were recruited from other pathways. And with this, I think I will go to my take-up message. A computer will always give you an answer, always. The hard part is not to get the answer from the computer. The hard part is to see which of the answers that you get have biological meaning and which ones are a result from zeros and ones and are not translating biology. The second thing is 16S diversity and microbial bioenergetics are two of the factors very important for diversity, but they do not necessarily co-evolve during the entire geological time. The history of our planet and microbial life is connected by bioenergetics. And I would like to end with an idea that I have that many people here might not agree, that microbial evolution and diversity is fooled by quests of different ways to make ATP and survive. And with that, I finish. Thank you.